Next up, I have the honour of introducing Dr. Tom Edwards. Tom is a vitreoretinal surgeon at the Royal Victorian Eye and Ear Hospital and principal investigator at the Centre for Eye Research Australia, where he works on developing treatments for inherited retinal diseases. So he's the guy to talk to get his work faster. So Dr. Edwards today will be sharing some insights into the research pipeline and clinical trials for IRDs. Thanks, Tom.
Thanks, Eden, and thank you and congratulations to the team here at Vision Australia. You do just a fantastic job, and I think I speak on behalf of all the speakers today that we're continually inspired by the work you do here and by all the patients and family here in attendance here today. Whenever I attend these events, I come away feeling with a sense of renewed determination to go back and work harder for you all in the lab and in the clinic. And it was interesting, also inspirational to see these memorial windows up here, which I was admiring while I was sat before, to Louis Braille, who I'm somewhat ashamed to admit I didn't know much about. So I was quickly Googling his story while I sat there waiting for the talks to start. And what an inspiration there as well. So he developed Braille when he was 15 years old. He lost vision as a 3-year-old messing around in his dad's shed and damaged one eye, and then lost vision in the other eye, probably due to an autoimmune sort of sympathy in the unaffected eye becoming damaged as a result of the trauma to the injured eye, and then was inspired to develop this system which was then named after him, Braille, and then sadly died when he was in his early 40s from TB. So, it's a shared journey for all of us, shared inspiration and motivation for the researchers, the clinicians and patients and families alike. And so, as I say, it's an honour to be here today. One thing I often think about is a really obvious question: why is the retina so vulnerable, seemingly uniquely vulnerable to these single gene disorders? What's the retina done to deserve this status? And I'm trying to remain humble here when I say that we research possibly the most complicated part of the body in the retina, and that's part of the problem. It is a highly specialized part of the body with many working parts which each require a specific gene to build the proteins needed for that particular part. So that's one of the reasons. Lots and lots of genes involved in lots of intricate little steps of these cells' function and structure. The other reason is that the retinal cells don't divide. Neuronal tissue doesn't divide. So that's why people who have had spinal cord injuries or strokes, for example, often don't do as well as someone who has had a broken leg or a nasty skin laceration. They don't repair themselves. They don't continue to divide after your embryological development. So that makes them uniquely vulnerable. And we often talk about how the eye is uniquely quarantined from the rest of the body in an immune from an immune perspective, making gene therapies an appealing, making the eye an appealing target for gene therapy. But that can make the retina more vulnerable because it doesn't have sort of other cells swinging in to support it when it is under stress or duress. And lastly, when Lauren was talking about rod-cone and cone-rod and this interaction between the different cells, the retina is uniquely susceptible to that, and a bit like a very delicate ecosystem. If one cell is affected, it can then cascade to affect other cells, which are highly interdependent. And then lastly, it's a hugely metabolically active part of the body with massive energy demands relative to its weight and size. And so that makes a bit like a nuclear reactor level sort of metabolic status. So small disruptions to the metabolism of the retina can cause toxic byproducts to accumulate and oxidative stresses to cause further cell death. So that's just a bit of background that sort of links into some of the talks you've heard already that gives you an idea of why the retina is uniquely vulnerable. But there are also advantages. It makes it uniquely also amenable to some treatments, which I'll talk about in my talk just now. But just a bit of background. So I'm an ophthalmologist. I'm very fortunate also to be involved in the research at CIRA and Cerulean with a fantastic team who you'll hear from today. So I do retinal surgery as well as involved in research. And so that research can be classified into different categories depending on where it's at in relation to it being an idea in someone's head through to it being an approved treatment. And so that continuum can be divided into preclinical and then clinical. So clinical meaning, you're applying it to patients or participants, and preclinical meaning not that, it's everything before that. And that usually starts, on a whiteboard or in someone's mind with an idea. And then usually you've got a series of experiments that are done in various models that represent some aspect of the condition or the treatment effect you're trying to bring about. So you'll do work in a laboratory with cells growing in a dish. They might be cells that have come from some random other part of the body and they're used just because they're easy to work with. They could be kidney cells, for example. And then you might have more eye-specific cells that you can just grow in a dish. It could be retinal cells, for example. And you'll do all sorts of tests depending on what treatment you're trying to do. So if you're trying to deliver a gene into cells, well, you'll do gene therapy on those cells. You'll put a virus that contains the gene of interest on those cells and see that you can get the gene in there. You can use special techniques to light up the new gene that's in those cells to see that it's there. And then you can do tests or experiments to see that not only is the gene in there and it's making protein, but is the protein it's making doing what it's meant to do? And so you've got different assays to determine whether or not the gene's functioning correctly. And then you've got a good feeling about that. Okay, we've got the gene in there, it's doing what we want it to do. Okay, is this actually going to work out in the context of an actual eye? And can we do it in a living creature? And often then you'll do animal studies, often in mice and things like that. And you'll basically go through that same steps. Can we get the gene in there? And is it doing what we want it to do? And often then you'll want to use an animal model that may naturally or have had a gene change induced in it to give it a visual impairment, for example, which you can then rescue. And if you've shown that, and you've got all that body of evidence together, and you can go to people with lots of money, because clinical trials cost a lot of money, and much more than the preclinical work. And if you can get through that massive hurdle, you can then make some of your product, if it's a gene therapy or whatever it is, at a much higher standard. It needs to be made to a level that makes it safe to use in humans, and that's one of the big costs. Which are usually borne by investors or pharmaceutical companies. And then you can go into these different phases of clinical trial. Now, this all sort of, we become familiar with phase 1, phase 2, phase 3, and it's even phase 4, but it's all still relatively new. This all sort of came in around the early 60s. The first sort of controlled clinical trials where you'll have, a group who have a treatment and a group who don't, and it's we're sort of familiar with that now, but that all came about in the 40s when they were testing antibiotics for TB around 1948 after the Second World War. And then, of course, that terrible tragedy of thalidomide, in the early 60s, really made the FDA in the US think we've got to have some more structure around this. We've got to prove that stuff's safe before we can just be, administering it to people. So this concept of phases, phase 1 being usually a small group of participants focused just on safety, and then phase 2 where you're looking more at early signs of efficacy, but also trying to work out the best way to test the efficacy, because you don't necessarily know what the best way is going to be. And that's a big challenge in our field. And then, if you've met, endpoints through those, phase 1 and phase 2. Then you're getting into phase 3, which are sometimes called the pivotal trials. This is the one where you want to actually know, does this stuff actually work? So you've often got a larger number of participants. Sometimes for inherited retinal diseases, the individual genes are often not that common, so they might need to be multi-center studies, around the world. And that's the result of the outcome of that. I mean, you can get all the way to that point and fail, as we've seen in some recent studies. But if you get a positive outcome from that phase 3 study, then you're going to the FDA or the TGA or the European regulatory authorities to say, all right, can we get this drug approved? And they'll base their decision on well, a lot of their decision will be based on the outcome of those studies. And then you've got phase 4, which I didn't mention here, which is when you've sort of got this post-approval analysis, and different things can come up there, like side effects that weren't picked up in the earlier stages. So to give you an idea of the sort of timelines, a good example to look at is Luxturna, which was the first gene therapy approved for inherited retinal disease. It's to treat rod cone dystrophy caused by mutations in a gene called RPE65. It tends to come on early onset, early childhood. So the phase 3 study was published in early 2017, and the FDA approved it in late 2017. So that bit's pretty quick, but there was a massive lead up to that with all the preclinical, phase 1, phase 2, all that stuff. That took a decade or so more of work. And then the TGA approved that in 2020. So keeping in mind this was the first gene therapy, but I was actually, the FDA approval was pretty fast off the back of that pivotal trial being published. Like, it happened in the same 12 months. TGA a bit slower, but still 3 years, that's not too bad. I mean, Australia's never approved a drug like this before. And subsequent approvals, I think, will be quicker because everyone's more familiar with it. The organisations and approval committees and people, they'll be like, oh yes, we've been through this before. So the outcomes they looked at and that were shown to be used to show efficacy were improved light sensitivity, improved mobility in a low light situation with a maze test. Had no major safety concerns. And so far, we've treated 6 patients at the Eye and Ear Hospital. We've done 2 just this year, actually, after a little break. And we've potentially got another patient we've just discovered through genetic testing. But like you heard from CC and Parker, it's not always straightforward. In fact, a good example of this is the 2 cousins we treated recently had the exact same mutation. I mean, it was in their family, but it wasn't one that we knew of before. It was one of these ones that, well, on paper, it looked like it would be a problem, but there were no other families around the world with this mutation that we couldn't automatically say, yes, that's we found the target. So then we had to spend about 2 years in the laboratory, basically almost like going back to the preclinical work. We had to make cells that had the same mutation, or we took cells from those patients and grew them in a dish. We had to show that the gene wasn't working. So this took a long time, but in the end, it was absolutely clear cut and the crispest data you could imagine. It was very clear cut that this was the cause, and they've gone on to have their gene therapy, and they're doing very well. In fact, I saw, I caught up with the male and female cousin, and they've both, one story was he has a modem or some kind of electrical equipment in his room, and it has one of those little LEDs on it that puts out a little bit of light. But he never knew it put out light. It sort of shone up on his wall. He'd never actually noticed that before. And since having, one of the first things he noticed was he was in his bedroom and he sort of opened his eyes and looked, and he could see all this green light going up on the wall, and he'd never actually seen that before. So there's little anecdotal stories like this that were quite inspiring. So I'm just going to talk about a few of the clinical trials that we've been involved with at CIRA/Cerulia. So, and just as an aside, so Cerulia is the sort of spin-out subordinate organisation that's come from CIRA to run all its clinical trials now. So they're intimately related, and Cerulia is still part of CIRA, really, technically. And clinical trials now get conducted through that arm of the CIRA family, if you like. A typo there, it's VISTA, not VIST-A. But this is a gene therapy. This is a phase 2/3. Sometimes they try and have it both ways, and they'll say phase 1/2 or phase 2/3. Clinical trial of gene therapy for X-linked RP caused by mutations in RPGR. So it's X-linked, so it's male participants only. That's not to say women can't also have a severe phenotype, even though they're technically a carrier, because that can be the case. But for simplicity's sake, in this trial, they're only including males. I understand there's 85 participants approximately enrolled worldwide. We've had 4 here in Melbourne. The recruitment is now closed, and we've treated some of our participants, and we also have some controls. The nice story here is an event like this I was at, and Lauren was there too, actually, in New Zealand a number of years ago. A family of a young boy who had X-linked RPGR-RP came up to me and was chatting to me just after a talk like this, and they asked me about trials that were coming in. I sort of mentioned this trial in its infancy. I was overseas when this was still at the preclinical phase, and I was telling them about it. And I said, "Oh, one day this will come to," and he's now in the trial, which is fantastic. So anyway, it's going well. The outcome measures are, again, they're having it a little bit both ways because of this issue of not quite knowing what are the best outcome measures to see early efficacy. So they're looking at vision under low lighting conditions, and also the sensitivity of individual retinal points in the macula, which can be assessed with a special machine. And so that study's underway, and I think this could be quite exciting. I mean, we've seen some positive early signs, let's put it that way. They should report on this towards the end of next year. They'll have some kind of press release. It probably won't be a formal paper that they publish at that point, but there'll be some kind of press release. So keep an eye out for that. So it's not all about gene therapy. In fact, gene therapy, its big weakness is that it is gene therapy for individual genes. And as Lauren told you just earlier, there's like now 400 or so genes. And it's just be an insurmountably impossible task to develop an individual gene therapy to all of these conditions. So there's likely to be another whole arm of therapeutics which are gene agnostic, and often they will be based on some form of neuroprotection effect that would work for any inherited retinal disease. And this is one of them, the Nacuity study. And this is a study that we've been conducting at Cira/Cerulean for a few years now. It's a phase 1/2 study of essentially an antioxidant medication with a proprietary tweak to it that makes it easier to be taken up into cells. It's called N-acetylcysteine amide, or NACA. The cysteine part of it is the key part, because that building block helps make a glutathione, which is an antioxidant. And an antioxidant is something that you think of oxygen as being good for you, but actually in some metabolic scenarios in the cell, oxygen can be harmful. And when you've got cells that are sort of thinning out and dying out in the retina, you've actually got too much oxygen, because the retina's got such a massive blood supply. You've got too much oxygen going through the retina. It sounds counterintuitive, and that can be damaging to the remaining cells. So you need a sort of a buffer in there, and that's what glutathione does as an antioxidant, and NACA is a few steps back in the pathway to make that antioxidant. Anyway, some really interesting and encouraging results have been published online recently. So they've shown that those on the NACA drug compared to placebo had a slowed rate of photoreceptor loss, and that reached clinical significance. They had a nearly 30% slower loss of visual function, which was a trend. It didn't quite reach visual significance, and it was safe and well tolerated. So this is really encouraging, because although this study, I should have mentioned at the start, was only recruiting patients with Usher syndrome, I don't see any reason why it couldn't apply to basically anyone with any IRD with an antioxidant neuroprotective strategy here. I think they kept it they wanted to compare apples with apples by keeping the same genetic inclusion criteria here. But as I said, this could be an exciting development. So this is another one to watch, basically. So another requirement for gene therapy is that there are actually some cells there to rescue. So if the retina has been so severely affected that the retinal cells, oh god, 1 minute remaining. Okay, better keep moving. The retinal cells have been lost, then you've got no cells to deliver a gene therapy treatment to. So that's where optogenetics can come in. So the inner retina, the sort of upper layers of the retina that receive signals from the photoreceptors that are typically mostly affected in RP, are all intact, and they're just sitting there dormant, or at least not getting the signal strength that they used to get from the photoreceptors. And you can grant cells, you can grant light sensitivity to these cells with proteins that will change their structure in response to being struck by a photon of light. And so the Abacus 2 study has just started at Cerulean. We've recruited 1 participant so far. These are participants with very, very poor vision. Even people who can't perceive light at all are eligible for this study. And it's not going to bring back their vision to what, is anything remotely like what they perhaps had earlier in their life. But the goal, that's not the goal. The goal is to give them some improvement in their vision, and that would be obviously very welcomed when you've got such a poor starting point at enrolment. So this is a phase 2 study. It's an intravitreal injection. It has to be administered regularly. It's not actually a gene therapy, so it's not delivered in a virus, and so it needs to be topped up. This photo switch needs to be topped up in the retinal cells. So this will be a good one to look out for. Quickly, because I'm probably running over now. This is a really exciting study that we've been doing for Stargardt's disease. So this is not a gene therapy, it's a tablet. It wraps up and binds to retinol, which is a key component of the visual pathway. We need retinol to be able to see. But the problem with ABCA4 associated Stargardt's is that the sort of byproducts of the visual cycle, which are normally cleared away, think of ABCA4 like the recycling van that comes around and picks up your recycling. It's faulty, so the stuff just piles up on the curbside. So that's a bit like what happens in Stargardt. So you get this stuff accumulating. So if you just buy less stuff, which is sort of how tinlarebant works, it reduces the amount of retinol going into the system at the start. You still need some, otherwise you wouldn't see, but it lowers the amount of stuff going in, so there's less toxic byproducts on the other end of the visual cycle. And if you get it just right, the vision's not affected, but it can reduce the flux, if you like, of these retinol byproducts building up, and therefore slow the rate of progression. It's not meant to be a cure as such. It's meant to put the brakes on it. Early results on this will probably come out next year. So keep an eye out for this one, the Dragon study by Belite Bio. So I think this is my last slide. So eligibility criteria was something that Eden mentioned that I should talk about. I mean, each study has different eligibility criteria. And as you've seen, so the photo switch Abacus 2 study, they were looking for people with very poor vision. Other studies will want people with better vision than that. You don't want people with vision that's too good, otherwise you're not going to be able to show a treatment effect. Then again, you don't want vision that's too poor. I mean, depending on the modality, that vision might be too poor to respond to the treatment that you're looking to see a response from. So with regards to eligibility criteria, really it's study specific. Traveling overseas for trials, this is an interest. I often get asked about this. You can imagine if it's a gene therapy which requires an operation, you need a Medicare number to have an operation in Australia. So if you're going to say a country overseas and you need an operation as part of that trial, then that might not be possible if you're not eligible to access that country's healthcare system. You've got multiple follow-up visits over a number of months to years. So you're really probably thinking about relocating to that country if you're looking at enrolling in an overseas study. I mean, some people might be able to do that. Then there's a whole sort of Pandora's box of insurance issues, and what do you do for your further follow-up with your local ophthalmologist? What if something goes wrong, if you have an adverse outcome? I mean, it is fraught. I'm not saying, it's not possible, but for a complex clinical trial that, involves, operating on the eye, that's a lot to take on. If it's something perhaps a little simpler, like some of these other studies where you're taking a tablet, like the Be Like Bio Dragon study, that's just a tablet, for Tinlareb's, it's a tablet, that might be more doable. I've already mentioned exciting results coming up in the next 12 months. Keep an eye out for those studies I talked about earlier. And as CC said, the first step to getting involved in clinical trials, or the easiest way, is to get involved with the Venture Registry. If you want more information on that, there's the email up there. So sorry I've run over, but happy to take questions later on. Thanks very much.